Welcome to Study Loop.
We are being asked to compare the regulation of gene expression in prokaryotes and eukaryotes. Let's start with prokaryotes. Prokaryotes have a circular genome, so they have a circular piece of DNA that contains all of the genes in it. RNA polymerases read those genes in that circular DNA to make RNA, and then the ribosome attaches to the RNA to translate it into protein. One interesting thing that happens in prokaryotes is that these two processes can occur simultaneously. Um, so basically, as the RNA is still being transcribed, a ribosome can attach to it and start translating it. Okay. Uh, the only regulation of transcription or of gene expression, the only regulation of gene expression that happens in prokaryotes is at the transcriptional level. So repressors or activators bind to prokaryotic DNA to modulate frequency and rate of transcription. So that is the only place where that is the only level where gene expression is being regulated to in prokaryotes, is at the transcription level. So you can imagine either repressors or activators can bind to the DNA here and either increase or decrease the affinity for the RNA polymerase to bind there. But then that's about it. In eukaryotes, it's a little bit more complex. You have a lot of different levels. Uh, so what I've shown here on the right-hand side is sort of the overall flow of information and levels of gene expression control in eukaryotes. So at the genetic level, or at the epigenetic level, so epi means above. So epigenetic, above the gene, and it's literally modifications above the gene. So you can add methyl groups to the DNA, and the addition of those methyl groups can result in a more tightly packed DNA, which is less accessible to polymerase. So that is at the epigenetic level. That is before the transcription level, before the transcription process is initiated. You can control gene expression in eukaryotes at the transcriptional level. The transcriptional level refers to the process where you go from gene to mRNA. And the binding of transcription factors to the gene affects that. Um, Once you have the mRNA can be translated into protein. And to the mRNA in eukaryotes, you can add these receptor binding proteins which modulate the stability of the RNA. So that is control at the translational level. And furthermore, in eukaryote, even after you make the protein, you can uh, you can affect how those proteins behave by the addition of other chemical groups such as phosphates. Phosphates are a common way to turn enzymes on in eukaryotes. So in the eukaryotic process of gene expression, you can regulate that at the epigenetic, transcriptional, translational, or post-translational levels. And the one answer choice that has all of those in there is choice A. Prokaryotes regulate gene expression at the level of transcription, whereas eukaryotes regulate multiple levels, including epigenetic, transcriptional, and translational. Let's look at why the other ones are wrong. Choice B, prokaryotes regulate gene expression at the level of translation. I don't need to go any further. Prokaryotes do not regulate gene expression at the level of translation. Choice C, prokaryotes regulate gene expression with the help of repressors and activators. That is true, whereas eukaryotes regulate expression by degrading mRNA transcripts, thereby controlling protein levels. That is not a wrong statement, but it is not a complete statement because the part about eukaryotic gene expression is not complete. That is only one step among the many steps where eukaryotic gene expression can be regulated. So choice C is not, it's less wrong than choice B, but it's not as right as choice A. Okay? Choice D, prokaryotes control protein levels using epigenetic modifications, and I can stop right there. That is not true. Choice A